This is part 3 of an introduction to virology and viral infections. In this section, I will cover the mechanisms by which viruses infect cells and replicate. There are 3 basic steps in which viruses infect cells successfully. Initially, they must adsorb to the surface of a target cell, adhere, and enter. Then they must reproduce their viral genome and produce viral proteins in which to encapsulate them. And finally, they have to assemble into a mature virion and be released into the environment. Taking the first step of this 3-part process, if we consider the mechanisms of viral adsorption and adherence to cells, looking at this from the point of view of the virus, there may be ligands on the surface of the virus that are synonymous with the capsid proteins, or there may be specialized spike proteins at the vortices of some icosahedral viruses that are designed to interact with the host cell. These are particularly common mechanisms in non-enveloped viruses. However, in enveloped viruses, surface glycoproteins are deployed into the envelope of the virus that are instrumental in attaching the virus to a host cell. From the perspective of the host cell, the virus may use a variety of different receptors. Usually these are functional cell surface proteins on the host cell, but they may be proteins that are limited in distribution to certain types of cells, such as growth factors or complement receptors or leukocyte markers. And these limited distribution receptors would account for the tropism of certain viruses for certain types of cells. On the other hand, there are other viruses that infect a variety of different cell types, and they may use functional cell surface proteins that are ubiquitous on a variety of different cell types, such as integrins or adhesion molecules. Whatever the cellular receptor is, they usually exist on the surface of the host cell in 10 to the 4 to 10 to the 6 number. And some viruses may require binding to 2 receptors on a single cell, and this actually increases the specificity of the virus for the cell because both receptors have to be present in order for the virus to enter. There are 2 main mechanisms by which viruses may enter cells. Some of the enveloped viruses will fuse with the cell membrane at the surface. That is, the viral envelope membrane will fuse to the cell membrane. And when this happens, it is a pH-independent process which delivers the nucleocapsid of the virus to the cytoplasm of the cell, as shown in this diagram. However, all non-enveloped viruses, and some of the enveloped viruses as well, enter the cell by inducing endocytosis. In this case, the virus, after it attaches to its receptor, induces the cell to take it up in an endosome, and typically acidification takes place, which may in some cases be the trigger that allows the virus to fuse with the endosomal membrane and then release the nucleocapsid into the cytoplasm. In some viruses, the proteins that interact with the host cell receptor are also modified to be fusion proteins. Either at the moment that they interact with the host cell receptor or via some other biological signal, the fusion proteins are designed to change their conformation such that the protein now forms a bridge between the viral envelope and the host cell membrane, and the protein is anchored in both. Eventually, the remaining domains of the protein associate with one another and create a motor which pulls the 2 membranes closer together. How they actually fuse once they become close to one another in apposition is still not completely understood. The second major step in viral replication involves the production of viral proteins and the replication of the viral genome. I will go through the major classes of viruses and indicate how this is accomplished for each of those. But understand that for RNA viruses, the events that we're going to discuss take place in the cytoplasm. A notable exception is influenza virus. For DNA viruses and retroviruses, the activities I will describe primarily take place in the nucleus. And a key exception here is poxviruses, in which Much of the action takes place in the cytoplasm. Positive-stranded, single-stranded RNA viruses require little modification in order to begin their replication once they arrive in the cytoplasm. Once the virus uncoats and releases the viral genome, since the viral genome is positive-stranded, it acts as an mRNA within the cytoplasm and immediately begins translating to proteins. Some viruses produce individual proteins from this translation, among which is the polymerase, which will ultimately assist with the replication of the viral genome. Other viruses, notably the enteroviruses, produce a polyprotein, which ultimately needs to be cleaved into individual products by a virally encoded protease. The virally encoded polymerase of which I spoke is an RNA-dependent RNA polymerase, a protein that is not found in human cells or mammalian cells. This polymerase will make a negative-stranded copy of the positive-stranded virus, from which multiple positive-stranded genomes can be synthesized. Ultimately, each one of these genomes will be surrounded by the proteins that have been synthesized inside the cell to create an intact virion, but this is something I will discuss later. Negative-stranded, single-stranded RNA viruses face the dilemma of having to be converted to a positive strand before proteins can be synthesized. The evolutionary solution to this problem is to produce a viral RNA-dependent RNA polymerase and include it in the infecting virion. So when the virus is opened to the cytoplasm, not only the negative-stranded viral genome, but the viral polymerase, which is contained and active from within the virion, is released. So the first step in replication of these viruses is to produce a plus strand copy of the negative strand genome. Again, once a plus strand has been synthesized, proteins can be translated from this just as if it were an mRNA within the cell. Some of these plus strand copies are also acted upon by the same viral polymerase to produce additional negative strands, which constitute new viral genomes. And again, these will recombine with the proteins inside the cytoplasm to produce intact virions. Double-stranded RNA viruses have a similar replication lifestyle. Most of these viruses, in fact all of them that are known, are segmented viruses, so each segment contains one gene encoding one protein for the virion. Once these double-stranded RNA genomes have entered the cytoplasm of the cell, they are acted upon by a viral RNA-dependent RNA polymerase, which is contained within the virion, and they synthesize a positive strand, which serves as mRNA, to produce proteins, each segment of the virus producing an individual protein. At the same time, the viral polymerase will also replicate the genome and make multiple copies of the genome that are double-stranded and available for packaging in a virion. Retroviruses have a very different lifestyle. They contain single-stranded RNA, and in particular, retroviruses contain two copies of their genome, both plus-stranded. When these copies enter the cytoplasm and are released into the cytoplasm, the first step is to reverse transcribe the virion into a DNA copy, and then to make a second strand to produce a double-stranded DNA copy of the plus-strand single-stranded RNA. Via the action of viral proteins which are present in the virion, the double-stranded DNA copy of the virus now enters the nucleus and integrates into the chromosome. And it is from this chromosomal copy that copies of mRNA are transcribed to produce the viral proteins, and additional copies of the plus strands are synthesized to produce viral genomes for packaging. With the exception of the parvoviruses, most DNA viruses in humans are double-stranded. So I will use that as the example to represent DNA viral replication in this lecture. Once the virus is taken up into the cytoplasm, the genome of the virus is transported into the nucleus and may or may not integrate in the chromosome. If it does not integrate into the chromosome, it may replicate as an episome within the nucleus of the cell. Viral mRNAs are transcribed from the double-stranded DNA genome just as they would be from host cell genes. However, DNA viruses are among the most complicated viruses that infect humans, and therefore with a huge number of gene products attributed to the virus, these viruses tend to sequence the translation and transcription of various viral genes. That is to say, the earliest genes that are transcribed and translated tend to be those that are associated with regulation of viral gene transcription. Then later, the genes that are associated with replication of the virus, maintenance of the virus in the host cell, and lastly, assembly of the virus into virions are transcribed in sequence. In most cases, the viral proteins that go into the final virion are transported back into the nucleus of the cell, where they assemble with copies of the viral genome that are replicated in the same way that host DNA is replicated. In many of these DNA viral infections, and cytomegalovirus is a good example of this, it's possible to see intranuclear inclusions with a light microscope, and these bodies represent clusters of viruses that are gathered together inside of the nucleus as they're being assembled. The final step in the viral life cycle is the assembly and release of the virions, and this can occur in two possible ways. One, through the deliberate killing and lysis of the host cell with release of virions from the cytoplasm, and the other way is through budding of the virus through the host cell membrane. In either case, the assembly of virions is a spontaneous event. That is, the proteins that make up the viral capsid assemble themselves, and they attract and assimilate the viral genome. In the case of budding, this process takes place at the cellular membrane, where it is organized. Most non-enveloped viruses and some of the enveloped viruses will use host cell death as a strategy for releasing virions into the extracellular space. In both of these cases, the virions accumulate inside the cytoplasm and then induce the destruction of the cell by a virally induced mechanism. Now, this could either involve necrosis, which means that the cell is damaged by, say, the inhibition of cellular protein synthesis or disruption of the host cell cytoskeleton, or alterations of host cell proteins producing a leak from the lysosomes into the cytoplasm. Alternatively, some viruses are capable of inducing programmed cell death or apoptosis. In either case, the host cell eventually disintegrates, which will allow the individual virions to be released into the extracellular space when this happens. Envelope viruses may use budding as a mechanism for exiting from the cell. This is a diagram proceeding from left to right that shows the various steps in viral budding from the cell surface. The budding process is initiated when viral matrix protein, which is produced in the cytoplasm, aligns itself against a section of the cell membrane. When this happens, the host cell proteins are pushed out of the way, and virally encoded membrane glycoproteins are put into the cell membrane instead, overlying the viral matrix protein. Eventually, the matrix protein stakes out a portion of the cell membrane and fills it completely with viral protein, excluding all of the host cell proteins. And when this happens, the nucleocapsid may then begin to associate with the viral matrix protein, and eventually the budding process occurs, causing the virus to exit from the cell while being surrounded by a cell membrane that it has modified. Ultimately, this virus can be released completely from the cell and becomes a free virion. The final panel in this section of the lecture shows an electron micrograph with viruses budding off the surface of an infected host cell.